Okay, you can go ahead and start doing your seating chart. And I'll pull up iClicker. Maybe I'll pull up iClicker. Wow, nice. So small. Okay, so don't forget iClicker and then your seating chart. So you're doing both. And if you're on Zoom, make sure you put your name down here where there's space for Zoom people. About 2 more minutes. All right, I got 2 jokes for y'all because Wednesday, y'all didn't get a joke, right? Okay, so why couldn't Cinderella play soccer? Because she was always running from the ball. All right, what did one eye say to the other eye? Just between you and me, something smells.
You already
Did I tell you that one? Oh, okay. All right. No, I've got, trust me, I've got plenty. Let's see. Okay. What do you receive when you ask a lemon for help? Lemonade.
I thought you said a lemon in my brain went to calculus and I was like,
okay, so make sure you're checked in on iClicker and on the seating chart. Now, before I move on, please go back and double check that you have the correct seating for for the exam, right? Because I just copied and pasted one of the days. So make sure it was right for yesterday. iClicker's not working. Try Yeah. Sometimes just give it till the end of the class period. It'll be open the whole time. So sometimes you just got to give it time. Okay, so make sure that your seating chart is correct for today and that the seating chart is accurate for Wednesday's exam. I am still grading the exams. I'm not the fastest exam grader, but I'll try to have them by Monday. But yes, ma'am. Yeah. So you can see. I think that's important. And you will not be able to keep them, but we will get to see them. All right. So we're starting a new chapter today. We're starting on chapter 4. I don't know what planet I'm on. All right. I want you to go and get something real quick on Moodle. If I could get to Moodle. Not textbook resources. So we are on material for exam 2, protein structure, methods, and function. So basically all of exam 2 is proteins. So if you look, there are the 20 amino acids. I would go and get that and have it kind of next to you for a while because you need to learn your amino acids, and we're going to talk about all of them, but kind of having that chart is really nice. And then on this same file, I have some, like if you want some ways to memorize them, there are a couple of different ways on here to memorize them. But to get started, having the table next to you is really helpful. Then you need to start learning them, especially if you are going pre-med. If you are going pre-med, you need to know your amino acids inside and out, left and right. I can't say that enough. You just do. So take the time now. There are a couple of free apps that are like flashcard apps that'll show you the structure, and then you say the name or things like that. But you need to get to know them. You need to get to know them very well. Okay, need the lecture notes. Here we go. Full screen. And I'm sorry, I'm supposed to put them on chat. Okay. Here we go. Okay, so chapter 4 is all about protein structure. So what drives protein structure? What determines protein structure? From the very beginning. What? Well, yes, they have certain structures to accomplish a certain function. That's true. But what gives a protein its shape?
The amino acids that make it up.
The amino acids that make it up. What determines which amino acids make up the protein? The mRNA, which comes from the DNA. So really, it's the DNA sequence that determines our protein's structure and function, right? From the very beginning. So if you have a mutation in the DNA, what's going to be a problem? Protein structure and function is not going to be right. Please y'all make sure masks are covering mouths and noses. Okay, so you can take any protein. For example, do y'all know about GFP? Like sometimes I like to start with a story. So GFP is the green fluorescent protein. It was originally isolated in jellyfish, but since researchers have been able to take that gene and express it in different organisms and get the same function. So the function of GFP is when you hit it with a blue light, it will emit a green light, right? So it's green fluorescent, it emits green. You can take it and you can put that gene into a mouse and you can make the mouse glow in the dark. How is that possible? Because you put the whole gene for GFP, you took it out of jellyfish, you put it into a mouse. You can do your cat, you can have a GFP cat. I mean, you know, it's all possible. All possible with science. So, but the point is that as long as you maintain that DNA sequence, you're maintaining the amino acid sequence, therefore you're maintaining the structure, therefore you're maintaining the function. Okay, so we're going to talk all about start to finish proteins. So what contributes to the three-dimensional shape of a protein? Well, what did y'all just tell me? the amino acids, right? So proteins are polymers of amino acids. So that sequence of amino acids is going to determine their 3D shape. Now, what happens, you know, all the proteins in your cells are not these long chains of amino acids, right? They're not these shoelace, you know, is what I'm kind of thinking, that you just stretch out and now this protein does its function. What does it have to do? It has to fold, it has to organize. So there are lots of things that go into conversion from a single polypeptide chain into a functional protein. And so all of the things in this entire chapter are all related to how you get a three-dimensional protein, a 3D protein, right? All of these things contribute to that folding. Okay, so start with the basics. Proteins are polymers of amino acids. How many amino acids do we have? 20 amino acids. It's a little abbreviation for amino acids, little a, right? Okay, so we know they're covalently linked together through amide bonds. What is the sequence of atoms in the backbone of an amino acid? NCC. NCC. Y'all remember that? I'm so proud. Yay. So if I wanted to link one amino acid to another amino acid, right? I have amino acid 1, amino acid 2. What's going to happen? I'm going to make an amide bond between this carbon, this nitrogen, right? So that's why you maintain NCC, NCC. So the nitrogen, what group is that? What functional group is that? An amino, which is NH, well, 3 positive or NH2, either way is right. So do you remember on the exam I asked you that? So what's the other functional group? Carboxylic acid, if you put the H. If you take away the H, what is it? Carboxylate, right? Carboxyl, however you want to say it. So you're forming the bond between this is my amino, this is my carboxylic acid. What do we call these? These are amino acids. Amino for amino group, acid for carboxylic acid. Are they named well? Yes, they are. Yes, they are. So cool. Okay. So when we talk about proteins, a string of amino acids can be really long or it could be relatively short. If it's short, we call them a peptide. If it's long, we call it a polypeptide. Okay, but it's still one string, like one shoelace, right? So peptides are typically less than 40 amino acids in length if you're talking about the short ones. So sometimes we even call those short ones oligopeptides. And it's just indicating that they're really short. So do you think a lot of proteins in the cell are going to be functional as oligopeptides or as peptides? No, they're really big, right? They're really big. So those are pieces that are going to come together to form a protein in the end, right? So you got to think about them almost as building blocks. So let's talk about this 20 amino acids real quick before we go on to the next slide. Let's just say I have this peptide that is 10 amino acids long. Okay, that's short, right? Proteins in the cell, 100 to 1,000 amino acids. Okay, so if I have this really short peptide, how many different variations of that peptide can I have? How do I do that math? Anybody know? You do however many, so at each place, how many different things could we have? Right, if I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Here's my peptide, right? And each of the X's is an amino acid. So at each X, how many different things could be there? 20. How many different X's do I have? 10. That's how you do the math. So that is 10 trillion possibilities. That's a lot. That's a lot. Okay, so if a short peptide could have 10 trillion different possibilities, why do we not have 10 trillion different kinds of proteins inside the cell? You have to have a certain combination to get a certain effect, right? So as much as there are possibilities, we are constrained in what those possibilities can do. If it is a arrangement that doesn't give us what we want, that doesn't give us a function, it's not going to be found in the cell. So it's not just a random organization of amino acids, right? We have to think about that these proteins are going to have a purpose. Okay. One of my tangents. All right, so when we look at proteins, why are they arranged in a certain way? And you have to think about function of those proteins, right? Where are they going to be functioning? How do they need to be designed in order to function in those particular environments? So for example, one of the weak interactions that was on your exam, if you chose to answer that question, was a hydrophobic effect, right? So what is the hydrophobic effect? It's that things that are hydrophobic want to get away from the water, right? So if you're in an aqueous environment and you have a protein, all those side chains that hate water are going to fold inward. So you have to have less organization of water around those hydrophobic areas, right? So if you look at, for example, a folded protein, each of these dots is an amino acid, and we basically separated them into two types. Either you're hydrophobic and you're yellow, or you're hydrophilic and you're red. So what do you notice about the hydrophilic residues? They're on the outside of the protein. But this is in an aqueous environment, right? That's really important to say because environment will determine a protein's properties. So this is one of the major driving forces for folding into a protein's three-dimensional shape, right? So we want these non-polar residues to be inside so that they can interact with one another instead of being forced outside and then having all this water have to organize around all these non-polar residues that are sticking out, right? So when you look at how a protein is arranged, you have to think about its environment. So if we look at these, I want you to look at them. I'm not going to say anything. I just want you to look at them for a second. A is one type of molecule. B is another type of molecule. Take a second and look at them and tell me what you think about them. Let me ask you this. What is the gray stuff? What are these gray things? That's the backbone, right? That's the backbone of the protein, right? Okay, what's the green stuff? Side chains. Do I have green? I think I do have green over here. Ha ha ha. So these are side chains. What about those side chains? Why do we have green side chains here and red side chains here? The green ones are hydrophobic. The red ones are still side chains, but they are hydrophilic. Okay. So now, this is one protein. Both of these two are the same protein. And this one, we're highlighting the hydrophobic side chains. Here, we're highlighting the hydrophilic side chains. Where are the hydrophobic side chains facing? Inside. They're facing inside. Where are the hydrophilic residues facing? Outside. Now look at protein B. Protein B, it's the same thing where this gray is the NCC backbone. The green are hydrophobic and the red are hydrophilic. Tell me about the green now. They're facing outward. And what about the red? That is completely opposite to what I just said. Why? They're in different, what environment is A in? Aqueous environment. What do you think B is in? Such as? Think about a cell of the membrane embedded in a membrane. Okay, so the first one, A, if you really want to know, do you really want to know? Yes, you do, right? A is called apolipoprotein. So apolipoprotein is one of those proteins that carries fats around in your bloodstream, right? Because you need certain things inside of all of the membranes of your cells and all that kind of stuff. And so this carries fats in bloodstream. So the bloodstream is basically what? Aqueous. Blood is basically aqueous. So you want to have something where your hydrophobic residues are facing inward, okay? This one is rhodopsin. That's rhodopsin. Rhodopsin is a transmembrane protein. Right? So if it spans the membrane, do I want my hydrophilic residues facing out? No, because they're interacting with nonpolar lipid tails. That would suck. I want to put them inside. So you have to think about the environment. We generally say most proteins are in an aqueous environment, generally, but not all of them. So it's important to make that distinction and to know what environment they're in. Then you can predict what the arrangement of the amino acids will be. Okay, cool. All right. I thought that was kind of an interesting way to look at it, right? Okay, so here are just a smattering of some proteins. All right, you have the protein name and what that protein does inside of the cell. Next to it are the number of amino acids that are found in that protein. Let's see if I can move this up. Each amino acid has a certain mass, right? And so we say the mass of one amino acid is equal to about 110 Daltons. So it's a unit of mass measurement. So if I pick something, let's pick something in the middle. Let's pick something that's maybe 500 amino acids. Let's just say that. I have protein X and it's about 500 amino acids in length. Can I predict the mass of that protein? Absolutely. And this is a really great tool that we can use in biochemistry, is to say, I know what my sequence should be because I have the DNA. So then I can predict the size of my protein product. And then I can do assays and does this bind to that? And if I run it on a gel, does it have the right mass? I can do that. And we'll talk about how we do that later. But how would I figure this out? Simple multiplication, right? I know one amino acid, we'll just say about 100 Daltons because it'll make the math really easy. What do we get? About 50,000 Daltons, right? Or 50 kilodaltons. So we can use that as a diagnostic tool. But what do you notice in this last column? Number of subunits in complex. Most proteins are super, super complex, which means that it's not usually one polypeptide chain. Usually, most proteins require more than one polypeptide chain folded together, linked together with lots of weak interactions, which ends up being really strong, right? Into a functional product. So for rabbit gamma interferon, how many individual polypeptide chains do we need? 2. For bacterial glutamine synthetase, how many do we need? 12. So what we call this is a subunit, right? So each polypeptide chain, each polypeptide is equal to one subunit. So most proteins don't function unless they have multiple subunits associated with them. Okay. And when we get to protein methods, we'll talk about how we can separate them, how we can identify them, and that's really, really, really awesome. So let's back up for just a minute and let's talk about amino acid structure. Right, so we know we have 20 different amino acids, and those 20 different amino acids are on that sheet that I was talking about earlier. And the way that they're different is by their R group, right? So if you look here, this central carbon, so we call this the central or the alpha carbon, same thing. The alpha carbon has the side chain attached to it. It also has the 2 functional groups attached to it. Those are the amino group and the carboxylate or carboxylic acid, however you want to think about it. And then that alpha carbon has a hydrogen attached to it. So sometimes this amino acid side chain can be really big, and sometimes it's really small. The very smallest one is what? Y'all know? Glycine. Why is glycine the smallest? Because its R group is what? It's just a hydrogen. So glycine is unique in the amino acids because it has 2 groups attached to it that are the same. So it would have this hydrogen and this R would be a hydrogen. So glycine is the only one that is achiral. I've talked about chirality before, right? Good. Okay. So glycine, because it's so small, it's a lot of times in proteins where you have a curve, where you have to pack things really tightly, all kinds of stuff like that. So you can think about what is the protein going to need to make this particular 3D shape, and then that's what amino acid is there, right? So this R group can be very different. Sometimes they have polarity, right? What if it has a hydroxyl attached to it, polar or non-polar? Polar, right? What if you just have a carbon-hydrogen tail, polar or non-polar? Non-polar, okay. So they can differ in polarity. There are some that are charged. Some are positively charged, some are negatively charged. Why would that be really important in a protein? What if that protein is supposed to bind to something else? Well, if you're supposed to have a positive charge in the pocket, the binding pocket, because your target is negatively charged, what happens if you have a mutation and you lose that positive charge? Now you no longer bind, right? So charge is really important, and it's usually in spaces where you have interactions occurring. Okay, so how does this all fit together for a particular protein? Well, however we arrange our polypeptide sequences in 3 dimensions is going to tell us what the function of that protein is. And there are a couple of different ways that you can get there. So let's look at example 1. Example 1 is an example of a heterotrimeric protein. What does hetero mean? Different. So what's different about this protein? You have 3 different polypeptide chains, 3 different. How do you get a different polypeptide chain? You start with a different gene, a different set of DNA sequences. So sometimes to make one protein, you have to start with different genes. They fold into a particular shape. Then these 3 will come together to make one functional protein. So each of these colored portions, we call it a subunit because it came from a unique polypeptide chain. And that gives this protein its particular structure and chemical properties. Another thing that can happen is that you have the same gene and you're going to make one protein from that gene, one polypeptide sequence. But to make the protein, this is non-functional. I need to associate one of those protein products with another copy of the exact same gene made into a protein. So I call this a homodimeric protein. Homo meaning same, di meaning 2, right? So I need 2 of the same to make one functional protein. Here, I need 3 different to make one functional protein. So you can look at the name and know what to call it, right? Okay. So a lot of times when we talk about the amino acids, when you're talking about an individual, it's not in a chain, we call it an amino acid. But once you start putting them together in these chains and you start folding them and they're inside that functional protein, we start calling them residues. Have y'all heard that before? People said that before? Not really? Sometimes?
I think that's the first time hearing that.
Okay, well, that's good. That's good. It's okay. So anywhere where you see residue. So a lot of times when they talk about mutations, it's residue 56, right? It just means amino acid 56. It just means that that amino acid is inside the chain, is inside the protein. Same thing. Residue, amino acid, same thing. Okay, so we talked about, oh, not what I want. Okay, we talked about our amino acids being chiral, except for one, right? Glycine's achiral. That's our exception. So all of your alpha carbons have 4 functional groups attached to them. The amino we talked about, the carboxy we talked about, the hydrogen, and then our R group. So if something is chiral, what does that mean? That they are non-superimposable mirror images, right? Remember that? Vaguely. And you can have the L orientation or the D orientation. And it's all about which side chains, which functional groups are attached to that central carbon and in what order. Because if I took this, and I don't think I actually can because it's all linked together. But if I took this D-alanine and I tried to just slide it over, would it match? It's like your left and right hand. If you slide them over, is it a perfect match? No, right? That's what L and D are. It's the same thing. Like your feet, your hands, those are all non-superimposable mirror images. So when you think about that, the 3-dimensional structure of proteins is super, super precise, right? There are only certain proteins that can do certain things because they're in the right 3-dimensional shape. How difficult would it be to have proteins that are in the L conformation and the D conformation in the same cell? Be extremely difficult because you would have to have one set of machinery to make the L and one set of machinery to make the D. Then you would have to have one set of proteins that would interact with the L and one set of proteins that would interact with the D. It's too much. Your cell would have to do double the workload, right? So you're not going to find both in a particular organism. You're really not going to find both basically anywhere. Okay. and that's not true. That's not exactly true. There are some in bacteria that do, but we're not going to talk about those. Okay, so if you look in your book, there's another designation as well. There's the S conformation and the R conformation, but unless you get really, really deep into biochemistry, you don't need to know them so much. All you really need to know is that most peptides, most proteins in general are in the L conformation. And if they're in the L conformation, typically they're in the S. And like, I'm not going to get that deep into it. I wanted to post, and I didn't get to it before today, but I wanted to post a little extra video for y'all on identifying if you're L or D and that kind of thing, because if you go to med school, you're going to have to be able to do that, but otherwise you won't. So I'm going to post that for y'all if you do want to go to med school and you want to know that more. Okay, so when we talk about these amino acids, I want to back up real quick. I want to back up to here. Okay. When we look at these functional groups that are associated with our alpha carbon, why are they charged? What's up with that? It's all dependent upon pH. Protonation state or attachment of hydrogens is all dependent upon pH. Okay, so how do you determine if something is protonated or not? You have to know the pKa. So the pKa is unique for each of the functional groups. So if I look at the pKa of my NH3+, right? My pKa here is about 2.3. If I look at my carboxylate group, this is, on the other hand, 9.7, much, much higher. So I want to know if I have a solution that's at pH 0, what are the protonation states of both the amino group and the carboxylate group? Amino and carboxylate are what? They're both what? So I'm here, I'm at 0. Protonated or deprotonated? Do they have their hydrogen attached? They're protonated because the pH is below the pKa. They're both protonated. Is that the picture I'm looking at here? No. How would this be different at pH 0? I put a hydrogen here, right? So this would be what my amino acid would look like at pH 0. So tell me, what's the net charge on this amino acid at pH 0? How many charges do I have? 1. Where is it? Amino group. It's a positive. So what's my net charge? Okay. Now, what if I take a pH value here? Let me erase this. Okay. And I say I'm at pH, I don't know why I did that, 14. Let's go like on the extremes. It should be, oh, it is. Oh, it is. Oh, 9.7, 2.3. Okay, I'm sorry. As I'm doing that. So you should have at pH 0, yes. So erase this, erase that. Okay. So if you're at pH 0, they're both protonated, you're going to have a net charge of 1, right? Okay, now go on the other side. Go pH 4. We're above 9.7. So what's going to happen when we get above the pKa of both of them? We're going to deprotonate. Both are deprotonated, right? So if I'm deprotonated, how does this picture change? So this amino group is going to be NH2. Am I going to be charged? No, that amino group is not charged. So how many charges do I have overall? Only one. Where is it? The carboxylic acid group or the carboxylate group. So what's my net charge? -1. So a lot of times when we talk about a protein, we want to know the pH at which the entire protein is 0. What? Or different? Yeah, but yeah, she was saying that. Oh, carboxylate. Yes. We want to know the pH at which a protein has a charge of 0. So that depends on 3 different things. It depends on the N terminus and the C terminus, right? That's what we just showed here. But sometimes that R group can have a functional group that is charged as well. And so it has its own pKa. So sometimes we need to know like how big our protein is. Sometimes we need to know what's the charge of our protein. All those things are really important characteristics that we have to know about our proteins. So when you're tasked with finding that, it's called the isoelectric focusing point. I'm going to get to it in the slide, but I want you to see it when you're looking here. It's going to be dependent upon not only the N terminus, not only the C terminus, but this R group. And so what we're going to do today is we're going to look at the R group of the different amino acids and look and see which ones have their own pKa values, which will contribute to the overall charge of the protein. Okay, so where were we? Here we go. All right. So sometimes when we talk about proteins, it's really hard to name each individual amino acid, right? You would be here for a really long time if you had 1,000 amino acid long polypeptide chain that you had to list the amino acids for. Your fingers would start hurting by the time you're done writing it. So we have 3-letter abbreviations and we have 1-letter abbreviations. Now you have to be sort of careful because there are some amino acids that start with the same letter. So for example, tryptophan and tyrosine both start with T. So the 3-letter abbreviation can start with T. So tryptophan is TRP, tyrosine is TYR. But when we go to single-letter abbreviations, we don't want to use a T, and we actually have a third one that starts with a T. So that's there. But what we're going to do is we're going to pick a different letter. So oh, where did I go? Here, tryptophan and tyrosine. So tryptophan and tyrosine, when you look at the name, if you make the same sounds, they will sometimes swap the 1-letter abbreviation for the sound that is made. So tyrosine has the Y, and tryptophan, I like to say tryptophan as if I were Elmer Fudd. Tryptophan, right? It's the W. I don't know, that's how I remember it, right? Right, and then you get the W. Okay, so sometimes it doesn't quite make sense, but the point is that we have more than one amino acid that starts with the same letter, so you're gonna have to find some different way. So you have to learn your 3-letter abbreviations. You have to learn your single-letter abbreviations, right? So I'm gonna put, what do you think I'm gonna put? Memorize. Flashcards, do something. Trust me, whether you go to graduate school, whether you go to professional school, you are going to need these. You are going to need these. All right. So when we talk about a protein, let's say, for example, we want to talk about a protein that's mutated, right? If I have hemoglobin, if my hemoglobin's mutated, what are some of the common disorders that are caused from defective hemoglobin? Sickle cell anemia, CK, sickle cell anemia, okay? How do you know you're gonna have sickle cell anemia? We look at your DNA sequence and we look at what proteins it codes for, right? And we look for mutations in the DNA sequence that lead to mutations in the protein sequence. So I'm telling you that to get sickle cell anemia, if you have an E6V mutation, you're gonna have sickle cell anemia. Tell me, what is that? E is glutamic acid or glutamate, depending on the pH. And I'll show you that in a minute. So that's E. So at residue 6, you normally have glutamic acid. But in the mutant version, you have valine. Okay, that's what that notation. So the first one is normal. The last one is the mutant. And then this is the residue number. Make sense? Okay. So in order to know, just by looking at this, if this is going to be a bad mutation or a silent mutation. Y'all know what a silent mutation is? Silent mutation is something where the DNA changes, but it doesn't change the amino acid, right? Or sometimes you can have a mutation where the amino acid changes, but it's close enough to the original that it's okay, right? Maybe it's not exactly perfect, but it's still functional. And so you don't actually have a phenotype. The person isn't sick. So if I just look at this, can you tell right now if this is going to be bad or not? Well, obviously it causes sickle cell anemia, right? But it's hard to know. So what's important to learn is the function of the different amino acids. What are their properties? How are they classified? And then you can tell if you have a bad mutation or not. Okay, so your book kind of groups them into 4 different groups. And while this is true, your book is not wrong, these are all true. Amino acids can fit in more than one category. Okay, so be careful with that. Amino acids can always fit in more than one category. So you have some amino acids that are charged. So aspartate and glutamate, these two, if you go and take a look at, let's flip over here. Okay, here are your aspartate and your glutamate. So these are charged and these are called acidic. Why are these called acidic? Look over here. What do we have? O minus. So what was this at a different pH? An OH, right? So that hydrogen can deprotonate. So that's acidic. Now I have some other ones that are charged, but we call these basic amino acids. Why are these basic amino acids? Or not necessarily if they have a double bond, it can be an NH2 with a positive as well. But it would be a nitrogen that carries a positive charge, right? Okay, so all my negative ones are acidic, all my positives are basic, and that makes up my electrically charged, or what your book says is that they are simply charged, right? So you can group these. You can group these as, what are my first two? Acidic. Acidic. This is so, oh, that is the wrong thing here. And acidic means it is negative. Lysine, arginine, histidine, what do we got? Basic, and they are positive. Okay. I like having one sheet where I can look at all of the properties of the amino acids together instead of having to flip through your book and all that kind of stuff. So they're all the same. All right, hydrophilic. In order to be hydrophilic, what kind of property must you have? What? Not necessarily. You could be charged. So if you are charged, now that's a great point. Because what did I just say? That these amino acids can fit into more than one category. So if you are charged, yes, you are hydrophilic. But we don't classify them as hydrophilic because they are charged and the others are not. So if you're uncharged, what do you have to be hydrophilic? Polar. These are our polar side chains. Does that make sense? All right. All right, so what about hydrophobic? These are nonpolar side chains. And I'm going to say that there's an exception in this list. Anybody know what it is? What is glycine? It's really the R group is really what? It's really hydrogen. So it's not that it's necessarily hydrophobic. It's really that it's inert. Right? It doesn't really do anything. It's not polar. It's not non-polar. That makes sense. What? It's just there. Okay. Then aromatic. So what am I looking for in an aromatic? Rings. So let's go back and look over here. So this sheet does not have your aromatics separated, but which three are they? Phenylalanine, tryptophan, and tyrosine. So if you want to like circle these on your paper and then identify these as, what do we call them? Now, why is this one aromatic not with the other two? Because it's polar, because it contains an OH here. So even though these all three are aromatic, not all aromatics are non-polar. It depends on what's attached to the ring. So you see how they can fit in different categories? Okay. All right. I'm going to switch back. All right. What's that? Good. All right. So your book, let me go back to view. Okay. So this is how your book kind of arranges them. And your book arranges them in a different way than what I gave you. Where's your NCC backbone in this chemical structure? See it, NCC? You have your amino group, your carboxylate. So this is your NCC right here. So then here's your side chain. When you look at the handout that I gave you, how is it different? And this, well, like I didn't do them, but here the side chains are doing what? They're pointing down, and here in the book, your side chains are what? They're pointing up. But it's the same thing, right? It's exactly the same thing. It's just pictured a little bit differently, okay? And here you're saying COO minus, but here, what are you showing? You're showing that this carbon actually has a double bond associated with it, okay? So I prefer this one, but you know, it's cool. Okay, so our charged amino acids, aspartate and glutamate. Why do we call them charged? What are we looking for? We're looking for what's going on in the backbone or the side chain? The side chain, right? Because if you were to say charged, I would look at each one of these and I would say, well, there's a charge on the amino group and there's a charge on the carboxylate group, right? But that's not what we're really referring to. What we're really referring to are those side chains. So if you look on these side chains are carrying a negative charge. When we draw all of our amino acids in books and all that kind of stuff, we are drawing them as if they are at pH 7. So pH 7 is between the pKa values of the amino group and the carboxylate group, right? So what's going to happen? The charge of the amino acid itself is not going to be so much dependent upon the amino group and the carboxy group. What are they going to be dependent upon? The pKa of the side chains, unless you have uncharged amino acids, right? Okay. So in this case, our ionizable side chain right here has a pKa of about 3.9. So if you're at pH 7, is it going to be protonated or deprotonated? It's above the pKa, deprotonated, right? Same thing here. This one has a slightly higher pKa of 4.1, but we're still at pH 7, so it's still going to be deprotonated. Yeah? Yes?
Can you say that again, like the pKa, like?
So here, so if the pH is above pKa, you're going to deprotonate. So here we're at pH 7. pH 7 is above 3.9, and therefore this oxygen is deprotonated. So at pH 7, aspartate is carrying a negative charge. Yeah. Okay. Because these charges, if this is positive and this is negative, they cancel each other out, right? So the amino acid would be neutral if there were no side chain that had a ionizable hydrogen. Yes. This is where those acids and bases information really get useful. Okay. Our other charged amino acids are what kind of amino acids? I like to box in my R groups. So these R groups are what? They're positive. What do we call the positive ones? Basic. Okay. So here we have an ionizable side chain with a pKa of about 10.5. All these pictures are at pH 7. So at pH 10.5, are we protonated or deprotonated? What? We're protonated. We have our hydrogen attached. So if our hydrogen is attached, what's the net charge on this molecule at pH 7? Positive, because here's a positive, here's a negative. They cancel each other out. This is a positive, so we have a net of 1, positive 1. Arginine, we have an ionizable at 12.5. We're still above pH 7, so we are protonated. Okay, now check this out. Histidine, this is really cool. Has a pKa of about 6. That's really close to what? 7. That's really close. So a lot of times it's the local environment of the histidine that determines if it is protonated or not. So it's really hard to predict for histidine if it is functioning as a positive or if it is functioning as a negative. So it's kind of one of those exception ones. So be careful with that one. Okay, hydrophobic amino acids. In serine, what gives me that hydrophobic characteristic? My hydroxyl, right? Threonine, what do I have? OH. Okay, in these, right, these are my side chains. These are super important because this OH can be phosphorylated. Do you remember we talked about that in the beginning? If something was phosphorylated and dephosphorylated, it can turn things off and on. Do you remember that? So if I have a protein and this protein is being phosphorylated, what am I doing? I'm taking a phosphate group from ATP and attaching it to my protein. Where do I attach it to my protein? On my residues that have a hydroxyl attached to them, right? These are the ones that can be phosphorylated. So inside the cell, we have serine and threonine kinases and phosphorylases. Phosphorylases that can phosphorylate and dephosphorylate, right? So that can take the phosphate on and off. And they say it here, right? So kinase adds, phosphorylase removes, right? Okay. Our other hydrophilic amino acid, cysteine. Cysteine is really, really cool because this sulfur group right here is super highly reactive. So if you have two cysteines that come close to one another, they are going to interact with one another and they're going to form a disulfide bond. A disulfide bond is relatively pretty strong. And so a lot of times, remember how I said you have peptide, if you have a homotrimer, homotrimer would work, right? That means you have 3 copies of the same polypeptide sequence. How do you hold those polypeptides together? A lot of times it's through disulfide bonds between the individual peptides. Okay, so I think we're out of time. Y'all have a great weekend. I hope to have your exams for you on Monday. Where do we stop? What slide is this? No, hydroxylic. Yeah, we're almost where I wanted to be. No problem. Thank y'all. Any questions on Zoom? Yeah, start learning your amino acids. Go get them. Please start learning those amino acids. All right, I'm going to end the Zoom. Maybe if I can get it.